Okay. Hello, everyone. Welcome to the Landcare Australia National Conference 2021 virtual conference. And our environmental and climate change stream resumes right now. Now, I would like to encourage everyone to submit your questions via the question box that you see on the screen. And our next speaker is Luis Morin, who's the research scientist at the CSIRO. So over to you, Luis. Please start your session.
Good afternoon, everyone, and welcome to this presentation on biological control of environmental weeds, which is an initiative supported by the New South Wales Environmental Trust. Now, before I start, I would like to acknowledge my colleagues, Hilary Cherry and Andrew McConaughey from the New South Wales government, who are part of the consortium overseeing this initiative. Also want to highlight the great contribution of the many research leaders that are involved in the initiative, and I have their name throughout the presentation, and you can see that then. So I'm sure that most people listening to this talk as part of the National Landcare Conference are well aware of the problems that weed pose to the environment. Now, environmental weed can be pretty detrimental in some contexts where they can threaten biodiversity and affect the functioning of ecosystems. While there are many different weed management methods available to control them, those methods are often not appropriate for environmental weeds because they are often too labor intensive and quite costly, or they may affect the non-target native plant species that we want to protect. So for the example, native plant species can be greatly affected by soil disturbance that occur when we manually remove weeds, or by the drift from the herbicide that we use to control them. So for these reasons, the development of biological control solution for important environmental weeds is quite attractive. So a new initiative to enhance research in weed biocontrol began in 2016 with financial support from the New South Wales Environmental Trust, and this is still ongoing. The guiding principle of this initiative has been to take advantage of previous or current research to reduce the cost, time, and resources to deliver new biocontrol agent for priority environmental weeds in New South Wales. So in other words, the principle has been to explore options to piggyback on previous investment made in Australia or elsewhere in the world to fast track the deployment of new biocontrol solution on the ground in New South Wales. So since 2016, the initiative has had 2 business plans comprising 3 different stages and a series of implementation plans. And the 2 key components of the initiative have been to do research on new candidate biocontrol agents, and also to release and evaluate recently approved biocontrol agent, and all this focusing on New South Wales. So before going into details on the activities under this initiative, let me provide you with a snapshot of what biocontrol is all about. This graph illustrates well the theory behind most weed classical biocontrol programs. So the red line represent the weed population, which typically fluctuate over time depending on environmental conditions. So when a biocontrol agent is introduced, so the blue line on the graph, it gradually builds up its population, which also fluctuate in response to changes in the weed population. So the severe damage caused by the biocontrol agent on the weed will gradually lead to a decline of the weed population below a so-called damage threshold, which is that black line in that graph. So below that threshold, the weed is still present, but it's not a major concern anymore because its population continues to be regulated by the agent below the threshold. So such a reduction in the weed population then opens up niches for colonization by desirable vegetation, which is a green line on this graph. And that's obviously providing that secondary invasion by other weeds are prevented to occur. So the key message here is that biocontrol agents do not eradicate weeds. Indeed, I mean, if they were doing this, it would mean their eventual extension from the environment, which would be counterproductive. So you see what I'm thinking here. There are many steps to implement a weed classical biocontrol program. So first, we conduct surveys in the native range where the weed come from in order to identify promising candidate agents, which are either insect or pathogens. The specificity of one or more of these candidate agents is then tested experimentally. And this is often done in a quarantine facility in Australia after we have imported those candidate agents from the native range into that facility. So if a candidate is shown not to pose a risk to other plant species, then an application for its release is submitted to the Australian government. And then once the agent is approved for release, it is then mass reared so that it can be released on a large scale across the range of the weed in Australia. I mean, this is often done in partnership with the community, such as Landcare groups. And finally, the establishment, spread, and impact of the agent are evaluated wherever it's possible, and there's resource to do so in order to assess really the benefits of the program. So here's an example of the benefits that was realized in one of the most successful weed biocontrol programs in Australia. So on the left is an infestation of broadleaf creeper that was smothering native vegetation in Yanchep National Park in Western Australia in the 1990s. Two biocontrol agents sourced from South Africa, rust fungus and a leafhopper, were introduced in Australia in the year 2000. Within a few years, especially in coastal area, the agent had a dramatic effect in reducing the weed population. So the photo on the right was taken at the same location as the photo on the left, but 3 years after the agent were released at that site. So in that short time, broadleaf creeper went from dominating the site to becoming a minor component of the vegetation. That was an impressive result. Now, it's quite important though for me to emphasize that not all biocontrol agents will achieve such an amazing result in such a short time. I mean, the impact of biocontrol agent may be more subtle, and they may also vary in time and space. So consequently, the integration of biocontrol with other control methods is often required and indeed generally recommended. So let's go back to the Weed Biocontrol Initiative supported by the New South Wales Environmental Trust. Now, the first stage of the initiative in 2016 consisted of going through a transparent process to prioritize the weed biocontrol target for investment in research on new promising candidate agents. So we started with a long list of widespread and emerging environmental weeds in New South Wales and identified those for which biocontrol research had been undertaken before in Australia or elsewhere in the world. We then assessed the prospect of biocontrol for these weed targets by determining the feasibility of starting a biocontrol project with an already identified promising new candidate agent. And we assess also the likelihood that this candidate agent would be successful. In parallel, we categorize each weed target on the threat that they pose to natural ecosystem. And we did that using an agreed weed risk assessment framework. A final matrix was then built, which is at the bottom of this slide, by combining the assessment of biocontrol prospect and weed threats. So this matrix was then presented to the advisory expert committee to the New South Wales Environmental Trust for them to recommend where investment in research should be made. So the funder here were making the decision as to, what level of risk they were willing to accept in investing. So since 2017, the initiative has invested in a wide range of research and extension activity. And I'm taking the opportunity today to provide a brief updates on some of them. Our specificity testing is ongoing with several candidate agents for a number of environmental weeds. Although some of these candidate agents may have been released in other countries, it's essential to test plant species native to Australia that are closely related to the target weeds to fully assess the risk before considering applying for their release in Australia. So the broadleaf pepper tree leaf feeding thrips, which originally comes from Brazil, is in the late stage of testing in both in Australian quarantine and in the laboratory of collaborators in the US, where the insect has been released in 2019. We will soon have results and will be in a position to decide if an application for release will be submitted. The small leaf privet lace bug originates from China and was released in 2015 in New Zealand. Its importation into an Australian quarantine for testing has been delayed because of travel restriction due to COVID-19. But as soon as we can get it in a quarantine, testing will first concentrate on an Australian native species in the same genus as the weed, because we think that maybe it could be susceptible, and we really need to determine if it is attacked or not. If the insect attacked this native species, then further testing will not proceed because we know we would never get permission to release the agent in Australia. The testing of the root feeding weevil that attack oxeye daisy in the native range in Europe is being undertaken by colleagues in Switzerland, as well as in an Australian quarantine. This promising candidate agent has not been released anywhere in the world so far, so it would be a first if we release in Australia eventually. An undescribed stem-boring herbivorous wasp was recently found on African lovegrass in South Africa, and collaborators there are doing initial host specificity tests to assess if this wasp has any potential for biocontrol in Australia. Now, this activity is part of a larger project funded by the Australian government under the Rural R&D for Profit program, in which the New South Wales Environmental Trust has co-invested. So we're trying to enhance activity by, investing in projects that are existing. A rust fungus that attacked balloon vine in South America was recently released in the Cook Islands. And from the Cook Islands, the fungus was imported in an Australian quarantine for host specificity testing in 2019. Now, unfortunately, the testing was recently discontinued after we found that this fungus can develop on 2 Australian native species closely related to the weed. Now, based on observation from the Cook Islands, this fungus would have probably been a great biocontrol agent for balloon vine in New South Wales if it had been shown to be sufficiently specific. Now, a key point here is the Australian government has very strict requirements before it approved the release of biocontrol agent. So based on the result that we've obtained, I mean, there was no option but to discontinue the research on this rust fungus, which broke my heart, I must say. Our specificity testing of 2 other candidate agents was recently completed as part of the initiative, and application for their release was submitted for assessment by the authorities. So a result from testing of the leaf cactus stem-wilting bug on non-target plants relevant for Australia, which was undertaken by a collaborator in South Africa, has formed the basis of the application to release, which was submitted in January 2021. An application to release a rust fungus for biocontrol of African buckthorn was submitted in late 2020, and it's still under assessment. The testing of this fungus was also part of that large project funded by the Australian government that I mentioned before, in which the Trust has co-invested. I should point out that our specificity testing of other potential candidate agents, which are insects, is ongoing in an Australian quarantine. So there may be additional agents that will eventually be part of the pipeline. The initiative has also include release and evaluation of recently approved biocontrol agent in Australia. So activities so far have focused on the coccinelle for Hudson pear biocontrol, which was first released in Australia in 2017. More than 33,000 coccinelle infested clados were produced for releases in the last financial year. So there's a lot of activity happening here. The initiative is also supporting the release and evaluation of the leaf smut fungus, which come from Brazil on wandering trad in New South Wales. In the last financial year, more than 200 releases were made across the states in partnership with the community, and multiple monitoring plots have been established at 14 sites, so we can evaluate over the next few years if there's any change, the damage that the agent caused on the weed. More recently, a fungus that causes disease on sea spurge, which is a beach invader, which was originally sourced from France, was approved for release in Australia in late 2020. The completion of all specificity testing of this fungus and the submission of an application for release, that was all part of the initiative in previous years. Since the fungus was removed from quarantine around Christmas time, the research has focused on developing efficient method for field release. Now, there's been new funding allocated for this as part of the initiative that just started this July, that will see over the next 3 years releases of this agent being made in sea spurge infestation in Tasmania and Victoria, which are the sources of seeds reinvading the beaches of New South Wales via ocean currents. So biocontrol can have this landscape effect that can't actually benefit much more than a single state once it's established and spreading.
Okay, now apologies, but thanks very much, Louise. Very interesting. You have 1 more minute. Can you please share some concluding points before we move to Q&A?
Exactly. Take home message. This was all planned. No. So to wrap up, here are a few points I would like to make. As you probably can appreciate, biocontrol programs require considerable upfront investment in research, and therefore sound investment decisions are quite important. And this is why for this initiative, we did all this prioritization at the beginning. safety of an introduced biocontrol agent is paramount. Now, this is why extensive research is done to demonstrate the specificity of a candidate agent before an application for its release in Australia is submitted. Now, the day of introducing generalist organisms like the cane toad will never happen again, trust me. Let's stop talking about cane toad in biocontrol in the same sentence. Biocontrol is not a silver bullet, and it will not eradicate the weeds. This is another important message. However, when biocontrol is successful, it can reduce the negative impact of environmental weeds in a sustainable and cost-effective way. And finally, if you want to keep up to date with progress under this initiative, now visit the website indicated on this slide or do a search with CSRO, weeds, biocontrol, and you'll eventually land in the right place. So thank you very much for your attention and interest, and I'll be more than happy to take some questions. Cheers.
Fantastic. Thank you, Louise. That was really informative. Now, Annie asked this. Some introduced grasses are highly invasive. What progress has been made in identifying species-specific control agents for grasses?
Grasses are a tricky one because we have a lot of native grasses, and to find agents that are sufficiently specific is always a big challenge. Now, I mentioned that there's activity on African lovegrass happening currently in the native range in South Africa, where they found a wasp that mined the stems that may turn out to be specific enough. So it's a question of watch that space. In case of other grasses, like, for example, buffalo grass or gamba grass, the problem we have is that there's portion of society that find those grasses helpful. They use in grazing, where there's another portion of society that thinks those grasses are actually quite invasive, and there's this dual confrontation. And because of this, we're still at that stage that is very difficult to build a case to go ahead with introducing biocontrol agent, and that's assuming we can find them. So it's possible that maybe the future of grasses will be we would introduce, let's say, biocontrol agent that either prevent seeding of those grasses so they're less invasive, or reduce the total biomass in the case of gamba grass in the north of Australia, so they don't fuel fires intensity as they currently do. So it is a challenge.
Okay, we have maybe one more question here. Stephen asks, leaf rust fungus was once successful in controlling blackberry infestations, but doesn't seem successful now. What do we know about how long biological controls work before adaptation occurs?
Okay. I've worked on the blackberry rust, so I can tell you, at the end of the day, these fungi that we introduced for biocontrol, they really work depending on the environmental condition. So, for example, in times of drought, these rust fungi are definitely not good. But then in recent years, for example, in the last season, with all the rain that we've had, the blackberry rust has been quite good in many places because it had so much rain. So it's not a question of adaptation. It's really a question of it won't work at all sites and it won't work every year, especially on blackberry, which grow in the summer, and that's when we may have really dry time. So as I said in the talk, it depends on the agent, depends on the weed, and it changes in time and space.
Okay, maybe one more quick question. Harden Landcare has been using biocontrol agents to manage Scotch thistle, noticing a huge decline in bugs that were plentiful across the area. Is any research being done to find out what happened? Just something quick, please.
Okay. Well, Scotch thistle, so you were saying that declining over time. I mean, the agent on Scotch thistle, they've been fantastic, but they've been knocked out big time by the drought. what we're finding now is that many sites, they're slowly rebuilding, but it will take a while. But it is one of those successful biocontrol programs. But guess what? It took about 20 years to actually realize how good these agents have been. And it's tree and second particular that work on Scotch thistle. So again, let's hope we have more wet years and then they will rebuild to as high population as they have been before.
Okay, fantastic. That was really informative. Thank you very much, Luis, and
Thank
thanks
you.
everyone for asking questions. And yeah, have a great day. And in 5 minutes, we will have another speaker at this stream. So if you want to stick around, yeah, stick around.
Bye,
Thanks very much.
everyone. Take care.